Back to part 2, where we will be discussing about bacteria. So when your mind hears about bacteria, you just imagine about some tiny creatures like and they are unicellular. They can't be seen through naked eye, so they are microscopic. As discussed in part 1, the same thing is repeated as we have already discussed about kingdom Monera. It includes the prokaryotic organisms. And talking about the history of bacteria, first of all, a scientist named Anton van Leeuwenhoek was the one who has seen bacteria for the first time. He was known as the father of microbiology and microscopy because he was the first to invent his own microscope. And talking about his academic background, he was a cloth seller person, and has he has used it for his business, the hand lens, and later he used a simple microscope. And eventually, one day, he took water through a pond and he observed it in his own microscope, and he was able to see something moving. And at that period of time, something moving means like it comes under animals. So he named those structure as animal kills. Animal kills, and these animal kills was coined by the scientist in 17th century to describe the bacteria, protozoans, and other small organisms he first observed in the water. And they were derived from the Latin for it is named as the little animals. And as we know that initial observation was made in the pond water, and it was in 1674. And later he examined it in his own dental plague. This is the dental plague. We can also it is known as tooth scum, and even in that he has seen something moving structures. And the later the term that is the formal name bacterium was later coined by Ehrenberg in 1829. And talking about the father of bacteriology is known as the is known that is the robot Koch is known as the father of bacteriology, as he also gave the Koch postulates. Likewise, the germ theory was also given by him, and the main thing is he proved the bacteria that bacteria are the cause of diseases like tuberculosis, like it is caused by Mycobacterium tuberculosis, like cholera, like it's caused by Vibrio cholera, like anthrax in cattle is also caused by bacteria. And we know that the bacteriology is the science that is the branch of science that is dedicated for the study of bacteria. This is the short history of bacteria. And talking about the largest, or we can say which is the largest bacteria, if it is asked, then from the historical view, it is in previous sections it was called as Bigiota mirabilis as the largest bacterial cell, which is up to 75 millimeter in diameter, and it is filamentous type of bacteria. And talking about the smallest one, it was called for the Dialaster pneumocyntis, that is the with the 0.15 to 0.2 millimeter. And according to the modern view, it is now known as the largest bacterium. If it is asked from the modern view, it is called for the Theomargarita magnifica, which has the sulfur pearls-like structures. You can see which can be up to the length of 2 centimeter in size. And talking about the smallest bacterium, it is for the Mycoplasma genitalium. That is, you know that Mycoplasma are the organisms that lack shell wall, so their structure, or we can say the shape and size also varies. So they come under the pleomorphic bacteria, that they can see change their shape and size according to the environment in it that thrives in it. And this is the same thing you can see. And talking about the characteristics of bacteria, their cosmopolitan distribution, that is, they are found in all types of habitats, like in hot springs, in deep oceanic floor, in deserts, or within the bodies of the plants and animals, and soil, in water, or everywhere. But it is not present in the burning fire. If you know that some active bacteria are also found in the volcanic vent, but the temperature, it is like double more hotter in case of the burning fire. So it cannot thrive in the burning fire. It is a simple structure with a unicellular structure. It's primitive, old structure, ancient structure. They are microscopic organisms. They have circular double-stranded naked DNA without histone protein. As already we had discussed in the part 1 about all this. And silver is made up of peptidoglycan, which is also known as murine, and somewhere also called as a mucopeptide, that consists of an acetyl glucosamine and acetylmuramic acid instead of cellulose. And they are both autotrophic and heterotropic mode of nutrition. They are the decomposers, natural scavengers, mineralizers in the biosphere. And basically the flagella may be absent in bacterial types like we can say in the cocci, like spherical bacteria, they lack flagella, and if the flagella is present, they are single stranded type. And talking about the vacuoles, they do not have a true sap filled vacuoles. Instead, there are gas vacuoles, like that helps for the aquatic bacteria to float. And likewise, talking about the type of ribosome, it has 70s type of ribosome, and it is prokaryotic organism, that is of 50 as that is a sedimentation coefficient 50 plus 30 is the combination here, where 50 is the larger unit and 30 as is the smaller subunit. And together it forms 70 type of ribosome. And the reservoir material in case of bacteria are the glycogen and lipids. And common mode of reproduction is binary fission. Usually sexual reproduction is absent. And then the next part we will be discussing the typical structure of bacteria. Thank you for watching. If you are liking my teaching style, you can just hit the subscribe button, and you can comment me the topics that you want an explanation from me.